summary as well as the results themselves. So I just want to go over this. This is in the lab documents module in Canvas. Okay. So I've got the results. It's a little Word document. You can see here. It breaks it down for Tuesday lab, Wednesday lab, for germination and shoot length between the 3 different seed types: radish, clover, and ryegrass. Okay. All right. I did a statistical analysis, what's called a student's t-test, comparing the control and the experimental control in alfalfa for germination for the 3 different seed types: radish, clover, and ryegrass. All right. We did not have enough data in the ryegrass in order to do germination, in order to look at the differences in germination. But just looking at it doesn't look that much, right? So it's actually not, there isn't enough replication to do a statistical test. So when you're writing your paper, you're talking about your results. So let's go back for a second and think about our null hypothesis. Remember what the null hypothesis, in general, what does a null hypothesis say?
Nothing happened.
Nothing happened, right? So there's no effect. And so we design our experiment in such a way that we have one treatment that's the control and one treatment that's the experimental, right? The null hypothesis says that the control essentially is the same as the experimental. Okay. If there is an effect, it means that the control is not the same as the experimental. Okay, remember that? Went back into this before? Okay. So what we're looking for are differences between the control and the experimental. And the term that gets used is significant. Is there a significant difference? Significant has a very specific meaning in statistics. It means that it is beyond a certain benchmark that you have set to determine if they're different or not, right? And for our purposes here, that difference is 5%, right? So they're 95, we're 95% confident that they are not the same data sets, okay? Because, just a little bit to back up, sort of what we're doing is we have, say, this is our control data, and it has some distribution. This might be a little bit, am I jumping ahead for people here? Does this make sense? So you have your average here, and you've got some measure of error, your standard deviation, okay? So let's say this is your control, and say this is your experimental. Okay, it's the average of experimental. We'll accept 5% overlap, okay? If it's more than 5% overlap, we're like, these are two different. So these are the same. If there's less than 5% overlap, they're two different things. Therefore, the control does not equal the experimental, and we can reject our null hypothesis. All right, you follow how the logic works on that? Yeah. Very good. So if you're doing, you guys are going to go in the medical field, okay? In the medical field, oftentimes it's not 5%, it might be 1%, it might be 0.5%. Okay? Now the way that, and this is because you really don't want to make a mistake, right? If, you know, the chance of getting cancer from this heart medication is, 5%, still way too high, right? We don't want that. So, okay, this is an arbitrary level. For ecology, 5% is fine. The way that this gets reported is what's called the p-value, so the probability, okay? So the p-value here, so for this first comparison, for the radish, germination, control, and alfalfa, the average is 29 for the control, 28 for the alfalfa. Our standard deviation is only 1. The value that comes out of the analysis is called the t-value, 1.2. This is degrees of freedom, don't worry about that. And this is our probability that they are the same. It's pretty high. That's a pretty high probability. You can see the averages are very similar. And when you look at the data, so when I plotted the data down here, they're pretty same, right? It's not that different, so it's not that surprising. So we'd say there's no effect here, right? So there's no effect of alfalfa on the germination of radish. Okay. Now on clover, on the other hand, average control germination 12.6, alfalfa 5.4. We still have this is a large standard deviation, but still our p-value is 0.018, 0.02 about. That's pretty good, meaning that indicates that there is a significant difference. Okay? So when we use this term significant, it's almost always in terms of a significant difference. Because what we're looking for is a difference between our control and our experimental. That indicates that there is an effect. Okay? So don't write that your data is significant. Data is always significant. It's your data. It's important, right? So what you will write is there is a significant difference, or there was not a significant difference. Okay? And when you write this, you have to include your p-values. Whenever you write, when you make a statement about your analysis, be sure to include the p-value. P equals something. Okay? All right.
What about the y-graphs?
Not enough. Well, there wasn't enough in order to do a statistical analysis, but you can look at them and say, it doesn't look like it much of a difference. Right? Yeah. Elena, I really need you to speak up.
Is there a minimum page count, and then in the minimum page count, like let's say it's 4 pages, is it do 4 pages of writing and then add your graphs, like, after the 4 pages?
I'm not a big fan of page counts, because some people can write very terse, and they can put it all in, and some people have it longer. I actually have a problem with you guys writing too much. Typically, that means that you don't really know what you're saying, and so you're trying to be too verbose. So if I say that it has to be at least 5 pages long, it gets me into trouble. So in the module, it has a list of how to write a paper. What I need to see is I need to see 2 graphs. I need to see your introduction, your materials, your methods, a results section. I do not want to see the raw data, and I want to see a discussion. Okay? And 3, we're excited. Okay? What's that? Doesn't matter. Doesn't matter. No. Yeah.
Are you expecting us to write out all of our observations?
All of your observations?
Or some of them?
So this is the challenge of writing a research paper. I'm expecting you to tell a good story. Okay? Yeah. So any good research paper is telling a good story. So if you kept track of the humidity and temperature for every single day that your alfalfa was growing, and that doesn't seem to have any impact whatsoever on your results, I don't need to see it, right? If, on the other hand, you were making good, clear observations, and you got some results that are kind of weird, it might be good to include those observations, because that might indicate something happened that you didn't expect. The time to bring that up is in the discussion. The place to bring it up is in the discussion. Does that help? You'll need an abstract.
Did we do it on all 3 of them, or just ones that you did?
All. Yeah.
So are we using the class data? Or
Yeah. Use your class data. Use the class data.
Can we like contrast between class
Sure.
data and?
And your individual, it's fine.
So we don't actually have to calculate anything.
I did it for you. Honestly, how many of you would have known how to do that? I didn't think so. So I just did it for you. I think it was kind of fun, actually. So, yeah. Yeah. What's that? So I have yet to decide if you guys are gonna do a second lab report. The second lab report is super different. It's this totally like weird, wacky thing that we do, and the analysis is, the type of analysis that would be necessary in order to actually do a statistical analysis is like way beyond the scope of what we would do in this class, so don't worry about it. Do you wanna do statistical analysis?
No.
You kinda wanted to learn, you're like, I don't care. You've done it before? You did in psych, yeah. Yeah, what's up?
Can we like write our like lab report and then bring it to you for feedback? Or
You're running out of time.
yeah.
I recommend, remember I told you go to the writing center?
Oh yeah.
So right, Melissa? So I recommend the writing center. If you get it to me, yeah, you're running out of time. I mean, you could give me something by Monday. So you guys have,
Say Friday, we can do all weekend.
so you guys have your exam next Thursday, right? I'm looking around the room at a lot of sad faces. Would you be upset if I bumped your exam back? Oh, no, that's fine. No, so we're running into a bit of a scheduling issue, right? So next lab, your lab reports are due. Next Thursday, you have an exam. The following lab, you got your lab practical. Right? So, you know, we could have the lab reports due next week in lab, and then, we can have the lab reports due next Thursday. That would give you a little bit more time to work on them. We would have the exam on Monday, then you'd have your lab practicals on lab, Tuesday or Wednesday.
Just leave it how it is.
Just leave it how it
The
is.
exam on Monday, next week Monday?
No, yeah.
Oh, yeah. Good.
Just leave it how it is.
I'm gonna leave it. I'm gonna leave it how it is. What's up?
I don't know. I don't like it either way.
How many points?
I'll go.
Amen.
Try it again. I'll go.
Amen.
The lab practical is worth as much as lab report, 50 points. Wait a second. So I wouldn't worry about the lab practical too much at this point in time because you've got next week to study for that. But basically, lab practical is going to be parts of the plant. So next week, we've done this a little bit. Remember, we looked at the parts of the flower, looked at the parts of the fruit. Next week, we're going to go through the models, look at parts of the model. We're also going to have to identify plants that we went through at the field trip. Yeah, yeah, plants.
When we have the lab practical and the lab report going same day, because like wouldn't that be less work? Like if we have just exam next week, so wait for just exams, then you have to do the report either way, which will probably help you study a little bit of the practical, maybe.
I don't think so.
Yeah.
I mean, it's,
I mean, unless you're gonna have to identify right where at the.
I think it's fine the way it is.
Make the lab report due on Monday.
Make the lab report to on Monday?
What's up?
This Monday?
Oh.
I'll go. Wait a second. Alyssa, so currently the lab report is due in lab next week. Alyssa wants to move it up to Monday.
Yeah, because then you have like 4 days to study for the exam.
I'm
and then
with
4
it,
days to
yeah.
study.
You, by the way, you can always turn
Maybe
your
a 4 days for the
you can always turn your assignments in early.
Yeah, I know. Apparently you guys don't have time management. So.
Oh, that was a cut.
You're so
Move it back. I wasn't moving back.
Gee, what do you think?
Oh yeah.
I know is we have our 212 exam on Monday, so
Stick with that, sis. All right.
kind of want to study for that.
I'll go. Lab report from the lab. Exam next Thursday. What's that?
Thursday.
We'll be doing we'll have a lab practical upon.
212 is
All right.
Tuesday. It's on Tuesday, not Monday. What's the point of crazy?
I'll go. Please look through this module I have here, how to write research reports, rules of thumb for writing research papers, how to cite t-tests in your results in your paper, t-test results in paper. And this is an example of a paper that a student wrote some years ago.
I would be doing
Okay?
some of that.
So read through all that to give you an idea. Yeah.
Where did he work the person mess up? Because you actually like deducted 5 points, but it's
Right.
not clear where is it.
It wasn't that good.
You just thought it was boring, so you
Yeah, it was
distracted
boring.
by
Yeah.
It has to be interesting, throw in like emojis.
You want me to like put a joke at the end?
No, so 45 out of 50 is good, right? That's a 90%. So that was a good solid paper, right? For one thing, they included the data. I don't want to see the data.
Can we include like a table?
I touched like two or three, 2.5. And the other 2.5 was because it.
So you're saying you cannot get
Points because it's boring.
Oh yeah, you can.
Like pretty unlikely.
What if we include like a table of data so then you don't actually have to look at it?
Why? I analyze it all for you. I don't need to see it again.
But like if someone else were to read it, they need to know what's going on.
You can always ask.
Nobody's gonna read.
So when you become a researcher, somebody will read your paper, and if they're interested, they'll be like, "Can I see your data?" They'll ask for it. Actually, putting it in the publication just takes up space. What's up?
So, we are going to use the class data, so
Yeah.
if, our like group data
Yeah.
is really contrary to the class data.
It won't be because it's all together.
Yeah, exactly, because like, in our group, like the radish.
Are you going to do your own analysis? Are you going to do your own t-test?
Nope.
Didn't think so. So you're not going to be able to really compare them. Right?
Thank you.
Yeah.
So some things like for our group, we
There's
got
less
like,
work to do. I don't. Yeah. What's up?
we got like molds and stuff.
You got moles?
The molds.
Mold. Mold.
Oh, you got mold.
Yeah, so are we just gonna ignore that fact? Yeah, because we're doing
What?
it based on the class data, and not everybody got
No one
molds,
got
so.
mold but us.
are we
So-
gonna-
We got mold. We got mold.
Tell a good story.
I don't know what stuff you're talking
I'll
about.
go. All right.
Me neither. No one knows.
I'll go. All right. For this, I need you guys to be clear and concise in your writing. Okay? I need you to be clear and concise in your writing, and I want you to tell a good story.
Open it.
So maybe it's like pretty creative. What if you take like you take out points and then they're gonna do
So some good rules of thumb. Numbers 1 through 10 should always be written out. Okay? Don't start sentences with Arabic numerals. No emojis. Always have a 0 before decimals. Never use the word very or really. Okay?
So we should use significantly and.
And when you use significant, it specifically means that your p-value is less than 0.05. Okay? Use the term significant to describe the differences and conclusions, not your data or evidence. Okay? Data is the plural of datum. It's a plural word. Okay? You know that? When reporting the results of a t-test, the p-value is more interesting than the t-value. You don't have to include the t-value. I've seen all the t-values. The p-values more interesting. People,
It's okay.
when they've written this before, they're like, the t-value, like, I don't describe the t-values. Because the t-value varies based on the size you're doing. Yeah.
For the worst percentage,
What are we supposed to
that
do
would be
without?
the test, do we need to cite?
For the t-test, you just put in parentheses what the p-value is, don't worry about it. When you're works cited, this comes into telling your story, right? So you need to do a little bit of research on what allelopathy is, how it works, you know, clover, alfalfa interactions, that kind of stuff. The works cited comes up in your introduction, your discussion. Those are the two places you see the works cited. Yeah.
Just to clarify, we're doing our lab report on all three of the.
All of them.
Okay.
Oh. That said, the ryegrass is kind of lame. I can't ignore it, but you know, we didn't get a lot of data on ryegrass. You guys didn't choose a lot of ryegrass.
9.
Now it's up to me.
Wow.
What's up? All right. Okay. Peter,
Do we get extra credit to make you laugh in the report?
Not necessary.
bring it closer to 50. All right. Look at this. You guys seen this? So this, I did. Oh, good. It's a sad flower. Okay, so this was the brainchild of Lydia Garris. You guys know Lydia?
Yes.
So what these are just white carnations placed in 3 different test tubes of a concentrated food coloring. Okay, and so what's happening here is transpiration, which we learned about last time. So petals are, I think I mentioned this before, they are modified leaves, right? So in the petals, you have the stomata. The stomata is where the evaporation is occurring from. As the evaporation occurs, the stomata, it is pulling the water up through the bottom of the plant, through the xylem. That is the cohesion tension theory that explains how plants transpire water from the ground up. Yeah?
So last time we were talking about this, the kind of impact on the roots. Is this supposed to be filtered through the water? And it's supposed to be filtered, so how come it gets the dot?
Well, a couple things. It filters to a certain extent, it can't filter everything. 1. 2, the roots on this were cut, and it was just the stem that was put directly in there. So the whole Casparian strip has been completely removed, right? So that's just not in the picture at all. But there is some filtration, right, that goes through. But even still, when I say filter, it's not a perfect filter. It's just a way that the plant can mediate some of the stuff that comes through because it has to actually pass through a cell in order to get into the xylem. It can't just go straight from the ground into the xylem. There is one point at which some type of filtration has to happen. Yeah.
So why is that when you have flowers in your house, like the water basically gets nasty and smells pretty bad?
Why does it get nasty?
Like bacteria or?
Yeah, there's stuff living, there's stuff living on the plant, right? There's a whole like microbiome on the plant itself. That's probably most of what's happening there. The other thing that happens is the water goes anoxic, right? So the water isn't exchanging much oxygen with the air, and so it'll go anoxic. And then once it goes anoxic, then you get all sorts of, microbes that start decomposing stuff. Types of things. Yeah.
If each of those flowers are on one type of dye, why are the petals multicolored?
Excellent question. So what's going on here is that, so what's happening, the dye is moving through, I have to say this is the root, it's moving through the xylem, goes up here through the stem of the plant, and then it gets carried into the leaves itself, right? And what's going on is the xylem is branching from here up into all these different branches into all these different leaves. And you can see by and large, it's pretty well differentiated, right? It's pretty well differentiated. But there are points at which it crosses over, because it's like streets in a city that intersect each other, that crisscross. And so the dye is coming out in different places. But it's pretty well differentiated. I mean, you can see the yellow here, the blue, the red, but there are places where in between where they do cross over and mix. Because because those two xylem tissues, pathways are going to the same petal. Yeah. Questions? So remember what's driving this is the difference in water potential, right? And the water potential is greater in the ground because it's wet than it is in the air. Therefore, water naturally, because of osmosis, wants to move from the ground to the air. The plant's just taking advantage of that. It's taking advantage of that natural flow of water from the ground to the air. Okay. If there were no tree there would just have the evaporation from the ground up. All right? Questions on that? Different colors? So each, the xylem, so here's the xylem, right? It occurs in a ring, right? So what's happened is this xylem over here is in one color, this xylem is in one color, and this xylem is in another color.
Isn't that all in one stem?
But they've been split. So remember, these xylems are tubes. They're cells stacked end to end that make these long tubes. So they're relatively separate from each other.
Yeah, but they're all in one color.
No, they're coming from three different tubes. Can you not see that?
No, I didn't see that, okay.
Sorry, I thought that was obvious.
Oh.
See that?
They're all coming.
Oh, no, they're coming from three different, sorry,
Is
I
the
thought you
stem
didn't
just
see
like
that.
split? Yeah, into 3 sections.
Yeah, they split the stem, they split the xylem.
Why can't you just,
Oh, that makes sense.
What's that?
why can't you just like put all the 3 dyes into one?
How could one color move like?
You could,
Yeah, but like.
and that's actually what, you know, I've done something like this in the past. This is why we hired Lydia, because she's way smarter than I.
Yeah.
I put them all in one, and you just get one flower that turns red, right, which is neat. But this illustrates the fact that those xylem are separate from each other. They're like individual straws that run up. Yeah. Yeah.
That means they cannot cross like exchange.
Not much. No, not a lot. No, there's some, but not very much. We'll look at that in a second. There's a little bit that happens, but not much.
So if you cut the half of silent or half of the tree, it's gonna die somewhere.
Yes, exactly. And that's one of the reasons why you'll see trees sometimes, you saw them on the field trip, where half of it was dead, but the other half was alive. It's because the xylem on one half was destroyed, but the other half was fine.
But it cannot like support the other half.
Right, it can't, it's hard for trees to do that, to grow, hard for plants to do that, to grow over that way. It's much easier for plants by and large to grow up and down than it is to grow sideways. They can do it, and you'll see trees do it sometimes, but if you totally break that xylem, actually it's the phloem that's also really important here, we'll get to that in a second, it's very hard for them to grow sideways. It's not, they're more inclined to grow up and down. Yeah. All right, so that is, okay, that's evapotranspiration. I want to talk a little bit about how the stomata work. So the stomata, remember, open and close. When they open, that allows the water to leave, that allows transpiration. Also, when they open, CO2 goes in. Now, what causes them to open and close? What they do, there is a chemical, a hormonal signal called abscisic acid, right? And when the stomata receive this signal of abscisic acid, they close off potassium pumps. And when they close off the potassium pumps, oh, I got that backwards. Oh, I got that right. They close off potassium pumps, that causes potassium to leave the cell, okay? When the potassium enters the cell, that increases the solute concentration, and then water flows into the guard cell that makes up the stomata, okay? So when there's potassium here inside of the guard cell, That reduces the water potential, water flows in. When the water flows in, that causes the cell to expand and become turgid. When it expands and becomes turgid, that is what opens the stone on it. Okay? So it's using water pressure as a means of opening and closing, and it's mediated by this hormone called abscisic acid. Plants do have hormones. Okay, we learned about pheromones earlier today. Hormones, as I've learned about hormones in the past, hormones are chemical signals that occur within an organism. They get transmitted between different cells within an organism. Pheromones occur between organisms. Yeah.
So this point is the potassium pump closed off. This is when the potassium is closed. This is when it enters the cell, so it's open. So potassium.
Okay. So the potassium pump.
K plus enters. And K plus
So the
leaves.
potassium pump turns, when the potassium pump is on, potassium enters the guard cell, okay? When it enters the guard cell, water follows, right? Water follows solute. That causes the cell to expand. That we call turgor pressure becomes turgid, and that causes it to increase. It receives this signal of the abscisic acid that shuts off the potassium pumps, potassium leaves, and then water leaves along. Okay. All right. Okay. All right, so we talked about transpiration. Transpiration is the movement of water through the xylem. Now I'm going to talk about translocation. Translocation is how sugars move. So this is occurring in the phloem. And the stuff that is in the phloem we call sap. Okay? There's a lot of water in there, but there's also a lot of other organic compounds, sugar, and then other things, we call them secondary metabolites. Think about tree sap, right? That's got other smelly stuff in there, antimicrobial agents that the plant uses in order to defend itself. So, whereas transpiration moves water solely from the roots to the shoots, in translocation, remember how the water, or how the sap moves? Source to sink. Good. Okay, and this, the way that we describe this is what's known as the pressure flow hypothesis. Pressure flow hypothesis. All right. Now, cool way that they figured this out. They used aphids. Guess what aphids are? Aphids are little insects. They're insects, they're pests, they're garden pests. They have a very long, what's called a stylet, basically it's a proboscis. It sticks the stylet into the phloem. The stylet is so small, it will fit into a single phloem cell, a single sieve tube, right? That's how small and tiny it is. Now, what they discovered was they stuck the stylet in, this is pretty nasty. They let the aphid stick the stylet in and then they cut it off. Right? I know. Feels kind of bad for the aphid, but whatever. And they cut it off, and then they were able to measure the pressure coming through the stylet. And what they discovered was that these sieve tubes are under pressure. Da-na-na-na-na, under pressure. Sorry. Wayne, very good. And David Bowie. Okay? So whereas in transpiration, xylem is under tension because the water is being pulled up, in the phloem, in translocation, the sap is being pushed. There's pressure in the system that they were able to measure using the aphid stylets. Okay? And here's how this works. So, at the source, and so the source can be a leaf cell, it can be a root, anywhere where you have a lot of solute. Chances are that solute is sugar. Okay, good. All right. The cells use active transport. Active transport means they have to use what? ATP. Now, if you've got sugar around, you can make ATP, because plant cells not only have chloroplasts, what else do they have? Mitochondria. Good. So there's plenty of ATP around, because there's plenty of sugar around. They pump in the sugar. It could be another solute, it could be something else, but most of the time it's sugar. Okay. Remember, water follows solute. Now you have all the solute inside of these sieve tubes, water comes rushing in. Where does the water come from? Comes from the xylem. The phloem always has to be located next to the xylem because it needs the water from the xylem in order to create the pressure, okay? So water comes rushing in, following the solute, now you've got pressure. This pressure pushes the solute through what we call bulk flow. And where does it push it to?
Everywhere.
It pushes it, good, it pushes it everywhere, but where does it end up?
The same.
The sink. It pushes it wherever the solute concentrations are the lowest. Where are the